and it's my honor to present here. So my name is Ming Tang. You can call me Tommy. I'm from China. So I will give you a brief overview of my career in my next slide. I'm currently the director of bioinformatics at AstraZeneca to develop novel therapeutics for cancer patients. Today, I'm going to talk about my unexpected career in bioinformatics. As you can see, the title. So I was trained in a wet lab, and I learned computational biology or bioinformatics the hard way. And I hope my experience can help you to avoid the mistakes that I made. So I will also share some practical bioinformatics tips and job seeking advice. And this is my Twitter. So I tweet mostly about bioinformatics, papers, tools, and tips. I also complain about it sometimes. So if you go and read my tweets, and this is my blog. So dive into geneticsandgenomics.com. And I have a YouTube channel called Chatomics that I started 2 years ago with over 100 videos so far. And yeah, go ahead and check them out if you like. So this is just a schematic view of my career trajectory. So I graduated from Shanghai Jiao Tong University in 2008. And then I started my PhD at the University of Florida. So I was trained in a traditional molecular cancer biology wet lab during my PhD. So everything was great. Okay, I had 2 first author paper publications in well-respected journal at the 4th year of my PhD. So not until like one day, year 4, when my advisor asked me to analyze this public available chip sequencing data that back in 2012. So I couldn't open it with Excel, so I was forced, to teach myself Linux, R, and Python. So I then did a 3-year computational biology postdoc with Dr. Wahak in MD Anderson Cancer Center, who was actually leading the TCGA, the Cancer Genome Atlas Glioblastoma project. So after my postdoc, I moved to Harvard Faculty of Arts and Science Informatics Division to work on single-cell RNA-seq and single-cell ATAC-seq data. So in 2020, I moved to Dana-Farber to lead an NIH-funded consortium project called Cancer Immunological Data Commons, or CIDC. And so that was a big project. So the data were generated from 4 cancer centers in the States and who carry out immunotherapy clinical trial. And they profile those patients using whole exome sequencing, TCR sequencing, RNA sequencing, and ATAC sequencing. So I was leading the team to pre-process all this data securely in the Google Cloud and do some downstream qualitative data analysis. So stayed there for almost 2 years, and then I joined a small biotech called Immunitas Therapeutics in Boston area. So where I built the computational biology team from scratch, and we use a single-cell RNA-seq and a single-cell T-cell receptor sequencing to do new target identification and making drug, this antibody, to help cancer patients. So currently, I'm leading the epigenetics bioinformatics effort at AstraZeneca. Okay. Okay, so this is me actually. This 2008. So I was trained, in a molecular cancer biology lab at the University of Florida, and every year we took a picture. So I think this is the first year. I couldn't speak too much English even at that time. I can't believe I've been here in the US for 17 years now. And then you can see I kind of gradually grow a little bit older. Okay, there are 2 gaps in my career. So the first gap is how I converted myself from a wet biologist, a bench scientist, to a computational biologist, right? And the second gap, which I am actually trying to fill in, is how to utilize my computational skills and data analysis skills for new drug development and eventually benefit the patients. So today I'm going to share my experience in filling the first gap here. So I had just a story published in Nature Career column. So embracing the command line, my unexpected career in computational biology. And this is the link that you can go to and read it for free, I think. And now it's behind the paywall. Kashin, I will share you the slides so people will get all these links from the slides. So before we get into what and how to learn computational biology, we really need to understand why, right? The answer is simply just one word: data, right? We're in the age of data exploding. So according to Sean Davis, he used to work at NIH. So the number of available bases in NCBI SRA as of 2018 is 1.8 times 10 to the 16, which is roughly 6 million human genome, Human genome is roughly 3 billion base pairs, right? And if you convert that to size, disk size of roughly 30 terabyte data was processed daily. And that was 15 years ago. Think about how much more data we are having nowadays. Okay. So biology has evolved to become a data-intensive subject with advancement of technology. For example, single-cell data, image data, spatial data. So it can be really overwhelming without data skills, right? That's why Florian titled this paper, "All Biology is Computational Biology," because biology has become a data intensive subject, right? However, in the end, he argues that computational biology is biology too, right? As you can read here, so pipette biologist, microbiology, microscope biologist, cell culture biologist, like you never heard about those terms, right? And they're all biologists. And computational biologists, they are also just biologists. It just happens that we are using different tools like computers. So I believe many of you work with data, and which category are you in? So I really like this cartoon I borrowed from Thorson, who is a bioinformatics professor in Australia. So if you know computer and statistics, and you also happen to know biology, in this domain knowledge, in this case biology, so you are a Superman or Wonder Woman, right? So I would argue actually bioinformatics and bioinformatician or computational biology, they should know actually all those three fields. Okay, I will use actually bioinformatician and computational biologist kind of interchangeably. There are some differences, but I'm not going to talk about the nuances. Okay. So what you should learn to tame the data, right? So we understand why, but what you should learn, right? Why should I just use Excel, right? The first reason is it cannot handle big data, right? Remember the first time when I started, I import this 2 GB data into Excel, it just crashed it, right? And this is just a different example actually, because this older version of Excel actually cannot handle like there's a 65,000 rows, and it caused the loss of this COVID data. So although the newer version of Excel can handle like much more rows, if you still have millions of rows, it becomes very slow and clunky using Excel, right? So the other reason is that using Excel is not reproducible for the results. And it's really error prone, right? In this example, so I think the authors, they mix up the rows or columns in the Excel, and then it leads to the retraction of this paper. Okay, more importantly, if you work with biology, Excel convert gene names to dates. I believe like more recent versions, they're trying to actually prevent this problem, but at least in old days, it's a big problem. And you see like for example, APR1 will be converted to April1, right? And Octa4 is a very important transcription factor that plays a role in like stem cells. Like it will be converted to October 4th. So next time, if you get a spreadsheet and you search your favorite gene, whatever that you're studying, and you cannot find it, it really might be because that it get converted to date. Okay, so be cautious with Excel. So Excel is still the primary tool for many users. I'm not saying that you should just abandon it, but to rather be aware of its weakness and the problems, right? Also, we need to follow the best practice. Okay, I highly recommend you to read this paper, Data Organization in Spreadsheet. Okay, you just Google it and you will find that paper. I think this should be the first course actually, like teaching taught actually in class and to the web biologist, but also to the computational biologist, right? So one of the best practices to make your data rectangular, okay? So we have actually two examples, like two versions of the same data here. The one on the left, which you have those like empty cells here, right? And so in this example, essentially you have different strains, like A and B, different strains of maybe bacterial. Then you have the normal strain, then you have the mutant strain. But in this experiment, you also have different time points, right? So you measure them, okay, at 1 minute, at 5 minutes. And I'm sure like I did this actually before, like so don't feel guilty. Like I did this before when I was doing my PhD. But the right way to do it is this way, okay? So every variable should have its own column. So you have a strain column, A and B. You have the genotype column indicating whether it's normal or mutant. And you have the time, minutes, it is like time column. So whether it's 1 minute or 5 minutes, then because you have also two numbers for the same actual condition, that's actually replicate. So you have the replicate number here, 1 and 2. Also, the last column will be the value, okay, the response. So don't leave actually empty cells, okay, in the spreadsheet. Okay. So go and read this paper. There are many more actually tips there. Okay, so what should you learn, right? So we talked about Excel. So you really need to learn a Unix command line, right? So we know humans and computers are commonly interacted with in many different ways, such through a keyboard, a mouse, like a screen, like a video like this where I'm talking to you. So this is so-called like graphical user interface or GUI, right? So with the GUI, we give instructions by clicking a mouse or using like menu-driven interactions. But the Unix command line interface or CLI is like a scripting language allowing like many repetitive tasks to be done automatically and fast, okay? So like for bioinformatics, the text file is still the key format, right? So Unix commands are perfect to wrangle those file formats. And most of bioinformatics tools, they're run on by the command line. And they're more efficient and powerful. For example, if you have a folder with a lot of pictures, but you also have a lot of other files. You want to copy all those files to a new folder called picture. So this is the new folder. And you can just use the CP copy command star. It's a white cards, anything. It will match any string, but end with PNG, and it will copy all of them in pictures, this folder. I mean, of course, the GUI way is like I will probably just manually use mouse to click off them, select them, then drag them into a new folder, right? I mean, it probably is doable for 10, 20 files, but if you have hundreds, like, I mean, it's just not possible. I mean, it's possible, but it's very tedious. Okay, lastly, like in like cloud computing settings or HPC or high performance computing clusters, usually you need to deal with the Linux commands interface, okay, command line interface. And I'm sure like for you guys who are taking this course, you have learned this terminal, right? So this is the command line interface, this GUI interface. And in this case, it's just a black box, right? But and with some text. It can be really intimidating, like in the beginning, okay, what's going on here? But if you just look at those two, they're the same thing. So those are just folder names in the home folder, home directory, and then corresponds into all the folders also on your computer, right? And most of the time, after you get familiar or comfortable navigating in the file system, go to different directories, things like that, and it becomes actually quite intuitive in the end, okay? So there's this one saying that Unix actually is user-friendly, but it is picky about who its friends are. So once you spend enough time there, it becomes actually second nature. Next, actually learn some Python, right? Python probably is the most popular language in data science. It's actually easy to learn and really powerful, right? In this cartoon, this guy said, "Oh, you're flying." No, I'm not sure how many people go to XKCD. I really like this website. Um, okay, you're flying how? And this guy said, Python. So this guy said, okay, I just typed import anti-gravity. That's it. Because there's so many actually pre-built libraries that are very useful. You can just use them and you can do many powerful stuff. So I am still a primary R programmer. And so R is a statistical programming language, which is free, open source, and powerful. It has its strength in statistics and also plotting visualization. So this is the IDE called RStudio. So if you use R, probably you know RStudio. So you can type actual commands there and evaluate them in the console, and then to give you the output figures here, or maybe some helping menus here, like documentation. Then here we'll give you some, for example, the variables in the environment. So this is R is actually very powerful. There are two books that I recommend: Advanced R. So those are freely available online, and Art for Data Science. Both are written by Hartley Wickham, who is the author of Tidyverse. Okay. So Tidyverse is a set of packages that you can use to wrangle the data, tidy the data, and plot the data. Okay. So this is a workflow of a data science project, usually. So this is taken from R for Data Science, the book that I just shared. And you import the data into R or into Python. Then you need to clean the data, tidy the data, because the real world data are rarely clean, right? They're all very messy. You have to clean them, whether there's some missing values, there's maybe some typos there, you want to fix them. And then, okay, you do transformation, visualization, the modeling, and repeat, and finally you communicate. But really, this tidying the data is a key skills that you need to learn, right? And the learning in Tidyverse is a very good investment, okay? And this free book is a good reference for you. And next, data visualization is critical in exploratory data analysis, or EDA. So before you do any downstream analysis, always plot the data, right? Get familiar with and look at data with your eyes. Okay, just use print commands and head commands and look at the first 6 rows, get all the column names, right? And data visualization is very important. Like in this example, all these different shapes, and they all have the same X, Y mean, and the standard deviation, and the same correlation. Okay, but they look drastically different. Okay. So you learn how to use ggplot2 as part of the Tidyverse package. So learn how to do data visualization and spend time playing with the data. So as you can see, it actually takes a lot of effort just to do all this data analysis, right? Tidying the data, plotting the data, and look at the patterns, things like that. But what people think we do as a bioinformatician, they think we have this magic keyboard that you can push buttons. Okay, I can just assemble the genome, I can call variants into RNA-seq analysis, make fancy figures like that, right? But this is a reality of a typical day of my life as a computational biologist. So I spent quite some time just installing tools, softwares, because there some tools are very hard to install. There are a lot of dependencies. And second thing is to Google. So I Google how to install it. Google if I get an error, like how what's going on, right? And then spend some time read the manuals of bioinformatics tools. Once you have the tools, and you need to learn how to use it, right? And that's actually a, like a criteria that I select bioinformatics tools. So if that tool, even it's published in a high-profile journal, but the documentation is very sparse, I'm not going to use it because I just don't know how to use it because I don't understand how the tool works. And converting file formats, right? Bioinformatics is notorious about different file formats, right? So just take, for example, whole exon sequencing as an example. You have FASTQ files. FASTQ is a file format. And then you align them into the genome, then you get a BAN file. So BAN file is a format. Then you call variants, then you get a VCF file, variant calling format. So it's a different format. So you see like there's just like so many different formats for bioinformatics, and then converting different file formats using different tools is a kind of essential uh, part of bioinformatics. And also, again, tied in the data, I already talked about it. And the real analysis, plotting, modeling maybe only takes like 20% time. Okay, so really, how do we learn things, right? Google is really how we learn and do things, right? In this cartoon, again, from XKCD here. So, but Rob, you use Unix, come quick, and if you can type this like correct tar command, so it's a command to decompress files. Okay, first try, okay, within 10 seconds, but then this guy failed. I'm so sorry because we just forget things, and really we just Google and find that command. Okay. And there's some actually Google tricks I want to share with you, right? And the first thing is, okay, you can use this bracket and R. So it will search R programming related pages. For example, if you search distance measurement and then square bracket R, so you will find actually only R programming language related distance measurement. Okay, not just, okay, how far like this city from that city, for example. And you can do that same thing for Python. Okay, okay, I want to learn how to rotate x-axis labels in Python using matplotlib, for example. And then if you use patchwork and bracket R, then you will find the R package. So you can use quotations to search for exact phrase. You can use the tilde sign in front of the word to find synonyms. You can use a minus sign to exclude the terms. And this is actually very useful. So you can search within a specific site using this format. So, okay, for example, if you search heat map and site colon, and then this URL, the bioconductor helping page, and it will search heat map inside that website. You can also define a file type. So heat map and then file type colon PDF. It will only give you the PDF files in the searching results, which is very handy actually sometimes. Okay. So ask for help, right? There are multiple places that you can ask for help for bioinformatics. So Seek Answer is one of the early websites that I go to. BioStars, Stack Overflow, and also the Bioconductor support site that I just showed you in the last page. And this is actually an old post that I posted in the Seek Answer website, I think 12 years ago. And you can see, okay, I was a beginner 12 years ago. I was asking kind of naive, stupid questions, but no question is stupid, but be proactive, ask and make an effort to ask good questions, define problem, what you have tried, what did not work. It's even better to have some code there so people can help you like better. Because one common thing that I see is that people didn't take some effort to ask good questions, think about it. If you don't take effort to ask good questions, don't expect other people to, like help you, for free, right? So you want to make other people's life a little bit easier. Always give enough context. What they have tried, what's the problem, what's the error message. Give like more information, right? That will help other people to help you. Okay. So this is the key takeaways, okay, from what I have talked so far. Learn Unix commands, Python and R. Google is the way. Now we have ChatGPT, embrace AI. So I find that ChatGPT is very useful to ask those questions, right? But the same thing, right? Be specific and give enough context. AI will help you better. And AI will not replace you, but it will replace somebody who do not use AI. Be cautious with Excel. So I didn't talk about this actually Git version control. This is a different topic that I'm a big fan of, reproducible computing. And the same thing for the project organization. So have a consistent flow structure for projects, reproducible computing. Learn by doing it. So the best way to learn something is to do a real project, either in your lab, right? Or like if you find some papers that it's relevant in your field, and you can download the data from the public domain, do that and try to replicate some of the figures in the paper. So I will have one slide talking about that. So that's more for your CV advice. Okay, but focus on your strength, right? Because you are a biologist. The biology domain knowledge is your strength. Okay. Okay, so now I will switch gears to talk about some career advice, I would say. So tip number one is to get on social media. I know people have different, perspectives for social media, but don't get into the trap of like politics, all this stuff. You go to Twitter, X, Mastodon, Blue Sky, or LinkedIn, right? Follow people of the same interest, right? And if you are interested in a particular field and you follow the labs and the professors who are doing good work, and these days, like in many bioinformatics journals, they have their accounts, so follow them so you get to know the most recent advancement of the field, right? The newest tools that was published. So I started using Twitter after I reading Stephen Turner's blog. He was the Bioinformatics Director at University of Virginia. And this blog is a how to stay current on bioinformatics. He has the 2017 version, also the 2012 version. I read the 2012 version and I started to use Twitter and other social media to follow bioinformatics. Okay. And second, this is actually some LinkedIn tip, okay? Make sure you have a professional picture for your LinkedIn profile. Use a related banner that people know what you're working on. The headline, it should show your actual expertise, right? Bioinformatics, like maybe metagenomics, like ctDNA analysis, for example, like depending on what you're working on. And this is small tip here, like I had this like target sign, this emoji in my name here. I use this to filter all this, robots, for example, like if I get a message from somebody calling me target Ming, something like that, because they use this automatic way to get your name, and I know it's from some bots, so I will just ignore them. Okay. The next step is write the blog. Okay, this sounds actually a little bit old-fashioned, old style, but this is from Yihui Xie, who is the developer of R Markdown and many other awesome packages. He said, "I web, therefore I am." So nowadays, if people, Google you or search you, and you don't have a blog or you don't have a website, you don't exist. Okay. So why do you need to have a blog, right? A blog post is much better than a statement. I'm good at R, I'm good at Python, I'm good at Bayesian analysis on your CV, right? So one practical tip of your CV is that show and not tell, right? Tell is like, oh, I tell you I'm good at this, but show is like, oh, I have this blog or my website link. Then people can go and see it, and you actually try to solve a problem, and you have code there, and essentially you should show them you are capable of doing that, right? Also, instead of spending like 30 minutes instead of spending 20 hours to write your CV in the last semester, you can spend 30 minutes every day. So for 5 years to build a website, right? So, and also there are many other things that are more suitable for web pages. Like, for example, the talks, the presentations, you can always put them in your websites, but usually I excluded them in my CV. Okay. So why a blog? Okay, this is continued. So this is a new blog post actually very recently in this year, and still from Stephen Turner. Okay, and he has this a new blog actually, and he wrote that okay, technical blogging is actually good for growth and also for own learning, in addition to the visibility, right, taking up space. Because like if you need to write down something to teach somebody else, you really need to understand it yourself first, right? So I always see blogging as a learning process for myself. So it's really I'm the biggest beneficiary, I think, for writing blog posts, especially technical ones. So build a website so others can find you, right? This is my website, diveintogeneticsandgenomics.com. I've been blogging for over 12 years. Previously, I was using a different platform. I think I was using Google Blogspot. And I think about 7, 8 years ago, I switched to Blockdown. So it's an R package. So you can use that to build website. Okay. And if you are actually not technical, you can use some other stuff. For example, Wix. Yeah. So my advice is start now, right? If you don't have a website yet. So the best time to start, one is 10 years ago. And the second best time is right now. Take a weekend just to set it up, okay? And you can use Blockdown, which I'm using, or the Squaddle. This is the next generation of R Markdown from RStudio. Or you can just use, Wix or those like easy to use services and to pay, I don't know how much money. It's like, but either way, I think it's a good investment, like for your future career. Okay. So lastly, make a GitHub repository. So as a computation biologist or as a bioinformatician, you got to have a GitHub repository and put your projects there, your code there, right? Let me talk more about GitHub. So as a hiring manager, first thing I do is to check if you have a GitHub link in your CV, right? You can put your course project there if you are allowed to. So if you are not allowed, I mean, if you publish a paper, I mean, that's best, right? So you can publish the code that is associated with the paper in GitHub. But otherwise, like if you really don't have anything, so I think replicating published genomic paper figures is a good way to show your capability, right? And this is actually my tutorial. Like you can find actually the link here as well. So in this website, I walked you through how to download the raw files, FASTQ files. It's from ChIP-seq experiment and do all this pre-processing step aligned to the genome, quality control aligned to the genome, and then call in pigs, make a big wig file for visualization, and then eventually remake the figure published in the paper. Okay, so it's a step-by-step tutorial. So yeah, make sure you go and check it out if you do want to replicate some figures. Okay. And third tip is how to connect with people on social media or in real life. Okay. So just this one core thing you need to remember is always give value to people on the other side. Okay. Always be a giver, not a taker. But you might you might think, okay, he's or she is far ahead of me. Like what can I do? What value can I do? I mean, you always probably know something that the other people don't know, right? So you can offer help on those things, right? Even if you don't, like take some effort, right? As I said, right? When you connect with somebody on LinkedIn, just don't click connect. If you really want to connect that one, add some notes, right? You can either also compliment, ask genuine like questions, like, be curious, and if you can offer some help, that's good. So for example, like interact with the LinkedIn post, for example, like it, like comment actually creatively. Don't just say, okay, congratulations. Don't just say, um, it's a good resource. Like, thank you very much. Like this, like just very low effort comments, right? But if you want to stand out, you always need to take some effort. Okay. So this is the last tip. Okay. So if you want to join a company, a biotech company or pharma company, I think pharma less so, but for a biotech company, those are the three questions actually my previous chief scientific officer, Thomas Tan, told me. So I'm listing them here. The first question is, are the science cutting edge? Are you excited about the science in that company, right? Second is also very important. Are the people you will work with kind and smart? Yes, kind first. So if they're not kind, they're assholes, like you don't want to go there because you're going to suffer and you're not going to enjoy the work there. And lastly is the finance of the company good, right? Usually biotech, the 5 year survival rate for biotech company, I mean, probably 5 to 10%, right? And they're constantly in the mode of raising new money, is in survival mode. So make sure when you join a company, it lasts at least 2 to 3 years, right? So you can spend enough time there, learn the new things, and then kind of accumulate some experience. Because I know people actually join a company and after 6 months they get laid off. It's kind of not good. Okay. Yeah. Okay. That's all I have. If there's only one takeaway, okay, from today's talk, if you forget all I have talk, it is actually believing in yourself, okay? Make the transformation you want. And you can see here, this is my actually tweet in 2013. That's 12 years ago when I was finishing my PhD and tried to find a postdoc position. I was tweeting, okay, is it possible for me to do a postdoc in bioinformatics? I only taught myself some R and Python. Okay. And you never know, like 12 years later, I can actually be in a big pharma company leading some bioinformatics effort. Yeah. So if I can do it, you can too. And yes, I think that's what I have here. And what questions do you have?
Some questions in the chat. I think one of the questions is back to the slide where you have the terminal and also the window with all the files. Someone asked why there is underline for the playground.
Underline for playground. Let me see. I didn't pay attention. Let me see. Underline. Let me see. Oh, I think I was using a different shell called OZesh. Oh my Zesh. Oh, okay. I'm typing here. Everyone. So if you Google oh my Zesh, you can find a GitHub repository. So there's underscore there because this is like a special actually configuration for Zesh. So when it's actually there's underscore, you can directly click it actually. So using your mouse, and it's just like an advanced feature of that configuration. Oh my Zesh, yeah. Anything else?
Yeah, and also for another question in the chat is there is a you showed us GitHub for the bioconductor website, the heat map. So when do you use that, or why do you need to search heat maps from the bioconductor website? I'm curious about the use cases.
Okay, yeah. I mean, you can make heat maps like using either R or Python, right? I think, but my actual favorite package is an R package called complex heat map. So if you Google complex heat map and then Google complex heat map and then bioconductor, you'll find that package. It's probably one of the most powerful packages out there to, you know, to make heat maps. And heat map is actually an essential skill for bioinformaticians, right? And then, but of course, also you need to understand it deeply. So I have actually other tutorials and links to explain what does actually a heat map mean. Okay, so let me see whether I can find it. Heat map. Okay. So I have it here. Let me send it to everybody. Some old stuff that I wrote like when I was learning. Okay, you see, 2016. That was actually early days. Okay, let me actually. There's another one. And the scaling. So when you're visualizing heat map, whether you want to scale the rows or do not scale the rows, and it's actually have very different looking for your heat map. Okay, so let me see. Okay, this one. And I have another link here. So make sure you actually read the last one actually. Then you can understand what is heat map actually deeply. Okay. Yeah. So the reason that I'm I'm searching bioconductor because I use complex heat map. Yeah, it's an R package.
Your favorite packages for produce research quality visualizations?
So you just learn ggplot. Learn ggplot, and you can make any figures. Like I actually made a video for that actually in my YouTube channel. Like, so if you master this, what, 6 or 7 different types of figures, because if you look at all those figures in the paper, like what are they? It's a bar plot, it's a scatter plot, it's a box plot, it's a heat map, it's a like line plot, it's a histogram. I mean, those are just basic things, right? Like if you know how to do that, and if you can wrangle, do pre-processing and wrangle data into the right format in a data frame, and you use ggplot to make any, reproduce any type of figures. That's how you learn actually the basics, the fundamentals, right? Then once you know that, you can, once you see the paper, you can kind of analyze, okay, what's the x-axis? What's the y-axis? What's the data? What's the plot type? It's a histogram. Okay, so then you know kind of what kind of data format you really need to reshape, and then to make it is ready for ggplot to make figures. Yeah.
Thank you.
Sure.
And Christian asking, what's the best way to learn and understand statistics in order to aid the study experimental design of a bioinformatics project?
Yeah, statistics actually is a. I always regret that I didn't learn 2 things like well in college. One is statistics, second is linear algebra. So those 2 subjects are kind of essential, like especially you want to like understand about experimental design or stuff. I actually wrote a very recent blog post actually. If you go to my post, and I have, let me send it here. I have actually links for learning in stats uh, and some like linear algebra there. Like nowadays, it's kind of amazing because there's so many YouTube channels. You can freely available, like high quality ones. Like StatQuest is my go-to to learn stats and also machine learning related concepts. And 3 Blue, 1 Brown I think that's it. They have the best actually video to explain linear algebra. Make sure you go there and watch. And I also have some books actually in the link, so make sure you can go and check it out. Yeah. Any other questions?
I think Thomas have a question. I think you might have addressed this earlier, but just making sure. I am a computer major, but I am interested in bioinformatics. Advice for me to advance in this path.
So you're a computer scientist, right? It's very interesting. I see this trend actually. There are many people from the other side actually. For me, is I'm from the wet lab, and then I learned computation. But there are actually a lot of more people actually coming from the other computer science actually background. They want to learn actually bioinformatics. So the suggestion I give you would be like learn enough biology, okay? And biology is very different from many other subjects. They are not written, there are rules, right? But the rules is kind of, there's always actually exceptions. For example, okay, a protein can play a certain role in this context, but it can play a different role, like it can be activating, it can be actually repressing in different contexts of different cell types, for example. So try to actually understand the biology. I can, if you're from actually computer science, in my GitHub repository here, let me send it to you. I have some links for learning biology actually. Hold a second. Um, learning biology. Yeah, there's some free resources. Okay, I just send it in the chat so you can go and learn more biology. Yeah.
And also mention for a low income learner, how are there any free resources? Because most of the courses are paid and very expensive. So are there any free resources, and can we leverage boot camps too? What's your opinion on the boot camps?
Boot camps, like this, I think you can start with my blog post, right? The bioinformatics roadmap. Most of the links there, they are all free. And there's some courses in Coursera, edX, and it's free too. Like if you don't need a certificate, it's also free. So most of them are free actually. Yeah. I think there's this saying like actually free resources are abundant on the internet. It is the desire to learn that is rare, or it is the action that you take that is rare. So if you take actions, there's always actually free stuff there. Yeah.
Is it necessary for one to get a PhD in bioinformatics in order to stay relevant in the field?
No, I don't think so. Like you, I, if you really want to be a professor, I think you do a PhD. But if you just want to do bioinformatics, doing data analysis, I think a master's degree is fine. And also, if you, I would rather you spend time, for example, in industry, in companies, and because PhD is like 6 years, and if you stay in a company, multiple companies for 6 years, you will learn actually as much as you can. Actually, those are all even more practical skills actually compared to PhD studies. Yeah.
Jasmine asks, you mentioned that after 6 months in biotech, someone can get laid off.
Yeah.
Do you have any advice on how to enhance your chances of getting the role, which is sometimes highly competitive? And do you have any advice on how to contribute value to a company such that you become essential to keep the company, for the company rather than getting?
You know, sometimes like, that's the company's decision when they run out of money. It's just like very hard for them to keep you. You know, it's like it's not your fault. You know, it's not that you are not good. It's just because the company doesn't have money, it's not your fault. So the best way to avoid that is to, okay, find a company at least it has a runway cash in the bank for another 2 years. And of course, you should be a high performer, right? Otherwise, they can lay you off even they have money, right? So, yeah.
What are packages would one learn at the start? Because there are a lot of packages out there for different purposes.
Yeah. So what exactly are you talking about? Like it depends on what data type, right? You're trying to work at different packages work for different data type. Like if you're working on mutations, you're working on, chipseq, epigenomics. So the way that I select packages is I go to, for example, it's a bioconductor package. You can go to the website and see how the rank, it has rank and also has a citation number, right? Like if you go to Google Scholar, find the ones that are well maintained with good documentation and a lot of citation, right? And also rank high in the bioconductor, for example. So it's popular and people are using that. Yeah.
Next question. Can one find a job with self-learning without a master's or PhD? Will employers in big pharma industry and research employ self-taught professionals?
Like there's, I mean, in big companies, usually it's harder. Like just the HR, they have independent, department HR. They will actually set some like criteria. Okay, you should have a master. Should have a PhD. Maybe for big pharma it's harder, but for small biotech, if I am a hiring manager, I don't care. I just go and look your GitHub repository. I will go and read your websites and your blog post and I see really you can do the job or not rather than a degree, right? So many people actually, like for example, Northeastern University here, they have a master degree program, which is 2 years, and I don't know, tens of $1,000 for tuitions. But many of the students who graduate with the master degree, they don't, they can't even do RNA-seq analysis from end to end, like from FASTQ files. But if you can show I can do this from the very beginning, using Google Cloud or whatever, using your Linux machine, I mean, I would hire you, right, rather than someone who cannot even do that with a degree. Yeah.
Question also asks, are there any paid intensive research internship opportunities for undergraduates and recent graduates that you know of that could be a help in their journey? I think.
Say it again. So undergrads, what's the?
So I think
internship, right?
Yeah, are there any paid intensive research internship opportunities for undergraduates and graduates?
Yeah, I don't know any actually, unfortunately. So that's why you should stay on social media, follow people, follow the universities. Maybe they have some programs, and you need to find them, right? You can ask ChatGPT, say, oh, who I should follow if I want to get information on those topics, like internship, and then go and follow them. And then when they have announcement, then there's an opportunity there. Okay.
And you can also set on LinkedIn to make them notification if there is any updates from that person.
Yeah.
Yeah, there is like an icon there. You can set it.
Yeah.
Akela asks, what advice can you give to foreign undergrad students who want to pursue graduate studies related to bioinformatics? No solid foundation background. Is it possible to get accepted for admission or scholarships? For context, I am a chemistry major from Philippines, and my undergraduate thesis is about immunoinformatics.
Yeah, take 1 to 2 years, prepare for that, right? Like if you don't have like solid foundation after 1 or 2 years, I mean, you will be amazed actually if you focus on that and how much you can achieve actually, and then manifest that and demonstrate that on GitHub, on your blog post. Then you have a better chance to get admitted to those graduate programs. Yeah.
Last question. Kay asked, I have a background in electrical and information engineering, but focus on optimization algorithms and machine learning to improve energy systems. I'm doing a postdoc using data science for clinical and microbiome data. How do I transition fully into bioinformatics? I use Python, C++, by the way.
So you're already doing kind of bioinformatics, right? Clinical data and the microbiome data. Like bioinformatics is a very big field, right? And you can always like subdivide into different subfields, like microbiome, metagenomics is just one of them. So you're already doing that, right? So because you have a like kind of electrical information engineer, so it's more like a programming background. So I would suggest you to just learn, right, biology, and then really depends on what you're interested in, right? If you're interested in analyzing next generation sequencing data, then okay, just do it from end to end, wrap FASTQ files to the figures, right? So that's how you learn it. And if you are really interested in algorithm development, then read those papers. Okay, what kind of problems are there, right, for bioinformatics? And you can, I mean, it just really depends on your expertise, right? If you want to do algorithm development, there are also a lot of labs that doing amazing work, just develop tools and algorithms. And for me, I'm more like a genomic data science. I analyze the data, but I use existing tools. But you can fill in that gap, okay, developing cool tools that other people can use.